Welcome to our connective tissue podcast. What we're principally going to do in our connective tissue discussion is focus on some of the cells that are part of the connective tissue. Now, as you see on the screen to the right, we have an entire collection of cells. We start out with a developmentally plural potential cell that is referred to as a mesenchymal cell, and we'll expand on that as we go. And this is going to give rise to a number of different cells of the connective tissue. Now, connective tissue, you usually think of being the collagens, the ligaments, the tendons, and we refer to those primarily as just connective tissue. But as you look at the cells on the screen, there are a number of different ones. We see cartilage, we're going to see bone, we see blood, we see cells of the immune system. These are sometimes referred to as specialized connective tissues, and for the sake of the cartilage and the bone, we refer to those as supportive connective tissues. But for our connective tissue proper, we're principally going to focus on the cells that you see in the box, the fibroblastic cell, the mesothelial cell, and to a lesser extent the adipocyte. These are the fat cells. They're specialized for fat storage. So that's what we're going to principally focus on with our discussion of connective tissue. And our objectives certainly are going to be defined the components that we find in the extracellular matrix. The connective tissue is the area between cells, so between an epithelium and an underlying smooth muscle, for instance. And so we're going to describe the fibrous components and the non-fibrous components. They're all molecules, but some are going to be globular, glycoproteinous cell adhesion molecules, whereas we're going to have our collagenous fibers, elastic fibers, and those are going to be the fibrous components. But most of what we're going to do, we're going to talk about the cells and distinguish the different cells of the connective tissues and understand the functions of some of these specialized connective tissue cells. So connective tissue has its name because it will interconnect various other tissues we have in the body and hold everything together. So epithelial tissue, muscle, nervous, you name it, that's going to be holding together. And you can see this collage in the illustration of our fibrous and molecular components and our cells that are interspersed throughout. This is typically what we're going to see in a connective tissue area of a histological slide. Now, we mentioned very early on that cell that was at the very top, the mesenchymal cell. This is an undifferentiated cell. That's a term we use in developmental biology for a cell that can become a number of different cell types. And we can go all the way back to the fertilized egg. The zygote is referred to as a totipotent. It's going to become every cell in the body. Then from a totipotent cell, we get to a pluripotential stem cell, which can become almost everything. It narrows down its choices. It can't be everything anymore. And then we get to a multipotent cell, and this is going to be, again, more narrowly defined, still a stem cell. It can become a number of different cell types. And that's what we're looking at with this mesenchyme population. In development, you have three principal germ layers, and the mesodermal germ layer that you see here is going to give rise to the middle portion of your body. That, of course, is where connective tissue is going to be derived, your cartilage, your bone, your blood. So mesenchyme are these multipotential stem cells during embryonic development derived from mesoderm that will give rise to our connective tissue components in the body. So our connective tissue is going to have a number of different functions. When we look at the pure collagenous connective tissue, we're going to see that those are arranged as ligaments and tendons. Those are going to be very, very dense parallel rows of collagen fibers, very few cells, little if any way of nerves, blood vessels, cells. So it's very strong. It's going to resist the stretching in your muscles to the bone connection and to bone connection. But we also see the bones and cartilage, that specialized supportive connective tissue that's going to support the body, allow for the leverage to work with the muscles to get work done. But secondly, we can think of connective tissue as a means in which we can have less dense areas so molecules can diffuse. Materials can pass from one compartment to the next, and so we say that connective tissues can be in areas where you can have exchange, and that's the liquid nitrogen tank. Please ignore that. It's not going to explode, I promise. Third, connective tissues can provide a place for defense and protection. And when we look at cells in the blood, the white blood cells, those are going to be part of our immune system. But certainly we can create a physical barrier with many of our connective tissue components. And then lastly, with our adipocytes, fat storage, that storage reservoir of energy that we can have for the body, that is all part of the connective tissue. So first, let's examine the extracellular matrix. This is the area outside of the cells, and this is an acellular compartment. And before we really began to appreciate the complexity of the matrix, a light microscopy term came into existence, and it was called ground substance, just basically a big word for stuff. And so the ground substance is going to primarily be proteins, carbohydrates, glycoproteins. And mixed within that ground substance, we have our fibrous component. And the two principal ones we'll talk about are our collagens and the smaller in diameter elastic fibers. Those are the molecular components. The cellular component, of course, principally are going to be the fibroblasts. These are going to be the cells that are depositing much of this extracellular matrix and maintaining the matrix as they migrate through this environment. So let's start at the ground substance, the non-fibrous component, and one of the constituents in the ground substance are molecules referred to as glycosaminoglycans, or GAGs is the abbreviation. GAGs are carbohydrate polymers, and they consist of a repeating disaccharide unit. You're going to have a hexosamine and a hexuronic acid, and depending upon those carbohydrates, you can classify them as either hyaluronic acid, which is an extremely huge organizing molecule, chondroitin sulfate, which is a rich constituent in cartilage tissue, and keratin sulfate. Now, of all of these, what I really want to point out to you is this sulfate group and that negative charge. That is going to be a magnet for polar molecules. It will sequester these polar molecules and positively charged molecules, and water, of course, is going to be one of those. So because of that negative charge, chondroitin sulfate is the molecular component in cartilage that attracts water, that gives it its resistance to compressive forces, and it causes it to be such a great shock absorber and barrier against friction. Now, if we take those glycosaminoglycans, yet we arrange them and covalently link them to a protein molecule, now we've created something called a proteoglycan. So on the far left, the black line that you see is basically a protein, a long chain of amino acids. And on some of these amino acids, particularly the serine residues where you can have an O linkage, we have attached either chondroitin sulfate or keratin sulfate chains, and there are other types of GAGs that you can attach, and you have created a single proteoglycan. Proteoglycans typically are going to be heavier in molecular weight because of the carbohydrate component than the protein. Now, we can add to our assembly and our structure of our proteoglycans, because if we then take hyaluronic acid, now in this illustration, the long line that you see that almost looks the same as our core protein, this is now a long carbohydrate polymer that's hyaluronic acid. And now each one of these side chains is no longer a glycosaminoglycan, but this is actually a proteoglycan. So you can see this little inset represents this proteoglycan. We refer to this as a proteoglycan aggregate. And hyaluronic acid can be so long, and you can have so many of these proteoglycans assembled, this aggregate of proteoglycans can be as large as an entire bacteria. So that is how big these get, and that's how rich this tissue, such as cartilage, is in those negative sulfate groups and the ability to attract water. Now, glycoproteins, on the other hand, are principally going to be protein that are glycosylated. And we add the adhesive term because these are going to function as cell adhesion molecules. They actually can cross-link the extracellular matrix with cell surface receptors such as integrins. One of those molecules we find in the basal lamina is laminin, hence the name laminin. And so when we look at laminin, it is a 3-chain protein assembled into a cruciate shape, and there are a number of binding sites. We can see that we have a collagen binding site, we have a proteoglycan binding site, intactin is another glycoprotein in the basal lamina. We have all these binding sites that anchor to the collagens and the extracellular matrix molecules, but then we also have a cell binding domain. This is where the integrin on the cell surface can grab laminin. It's anchored into the extracellular matrix, and so that is how we have the cell adhesive function of molecules such as laminin. In cartilage, we have a molecule with a similar function called chondronectin, bone osteonectin, and then secreted by fibroblasts that is basically very widely distributed throughout tissues, including the basal lamina and reticular layers and just other interstitial connective tissue. We have fibronectin. That was the first one that was really described. And when you look at fibronectin, laminin, of course, as we see, has 3 chains. Well, fibronectin is going to be a dimer. And these dimers are linked together. And once again, in this illustration of a fibronectin molecule, we can see a collagen binding domain, fibrin binding domain. Remember, fibrin's important in clot formation. Heparin binding domain. This is another glycosaminoglycan in the extracellular matrix. But then we also have a cell binding domain. And you notice within this blue cell binding domain, we have this red stripe that says RGD. Those 3 amino acids are critical for the ability of integrins to recognize and bind to many of these cell adhesive molecules. And so that RGD sequence is your textbook typical 3 amino acid sequence that represents a cell binding domain. If you change any of those 3 amino acids, fibronectin loses its ability to interact with integrin receptors on the cell surface. Collagens now, as we move into our fibrous components, collagen is the one that we're going to spend much of our time with as we talk about connective tissue, especially our collagen type 1, our collagen type 3. These are our interstitial types of collagens. We also talk about collagen type 4, which is present in our basal lamina, in that lamina densa region. But collagen is a protein, and its function is to confer tensile strength. Now, that is to prevent stretching. Elastic fibers that we'll get to causes a rebounding when a tissue is stretched. Collagen is going to make sure that this tissue doesn't stretch too far. And so they're going to somewhat have cooperative functions or antagonistic functions, collagen and elastin. But collagen is going to be this protein that is arranged together into a pro-collagen molecule. Now, that it consists of 3 protein chains that are basically helices that are braided together. And they have leader sequences that are going to cause them to hold together long enough to form this 3 helical structure until we reach a point that enzymatic degradation is going to remove both ends of our pro-collagen molecule, and that will result in a functional tropocollagen molecule that is now ready to be assembled into a collagen fiber. So as we look at our illustration, we can see our 3 helical chains of collagen. They're going to be assembled into a pro-collagen molecule. We will chop off the ends. That will be our tropocollagen, and then we will assemble our tropocollagen molecules into this array that is going to be our collagen filament. I don't like this term microfibril. We're going to reserve that terminology for something else. We're just going to call this a collagen filament. Now I want you to notice, you see the cross links between the collagen molecules, and these cross links are going to result in a staggered appearance of the collagen molecules as they are assembled. That is going to result in what is referred to as a periodicity to the collagen molecule, essentially a striped appearance. And so when you look at a transmission electron micrograph or a very high power scanning electron micrograph, you're going to see these very regularly arranged lines or these grooves on the collagen filament. That is periodicity. Do not confuse this with striations in striated muscle. These are going to be much, much smaller in diameter than we'll see with our muscle, but the stripes are the periodicity of the collagen molecules. Now, the cross-link that we have to tie those tropocollagens together are mediated by the amino acid lysine. And they're going to create a bond that's referred to as a hydroxypyridinium cross-link. Now, not being a biochemist or a chemist, I really didn't know what a hydroxypyridinium cross-link was, but this is what it looks like. Your two adjacent lysine molecules are going to join together, and they are going to remove the amine groups, link them together, and this is their hydroxypyridinium cross-link. Collagens are rich in this lysine, so you're going to have those cross-links throughout the collagen filament, tying those tropocollagen molecules together. Now, as of the time I put this together, there were more than 15 known types of collagens just simply numbered together. Here are a few of the more common ones. Collagens type 1 and type 3. These are going to be your connective tissue type, and collagen type 3 is sometimes referred to as reticular fibers. I refer to 1 and 3 as connective tissue collagens or interstitial collagens, so you'll hear me refer to them as such. Type 2 collagen is characteristic of cartilage. However, we realize as of the last, I'd say 10-15 years, that collagen type 2 does occur in other places, but we still associate type 2 with cartilage. Type 4 is our basically butterfly mesh-like arrangement of collagen, and we find that present in our basal lamina principally. Which lamina do we find that in? The lamina densa. Type 5 is a placental type collagen, and then we saw our type 7 collagen, the interconnecting form that interconnects the collagen 4 of our basal lamina to the collagens 1 and 3 of the lamina reticularis. Now, as I said earlier, elastic fibers are almost the antithesis of collagens. Collagens resist stretch. Elastic fibers don't mind being stretched, and they have the great ability to rebound quite easily. Now, elastic fibers are proteins, and they may be stretched to 1.5 times their resting length. Now, elastic, like collagen, is synthesized by fibroblasts, but smooth muscles also have the ability to produce elastic fibers, especially the ones that are associated with blood vessels. We'll see a lot of elastic lamina around blood vessels, especially arteries. They have to resist the higher pressures of the blood flow, and so that elasticity is going to allow them to stretch and rebound back and confer a little bit of pressure on the downward cycle during our diastole of the heart. Now, elastic monomers are joined together into fibers as well. These are going to be smaller in diameter than we saw with the collagens, and they continue to use lysine to make these cross links. However, we are going to join 4 lysines together, and those cross links are going to be called desmosine cross links. They're going to hold the elastic fibers together, not in so much a regular linear array, but more of a lamina and a sheet, or maybe even a belt, if you want to think of it like that, in many of our organs that we're going to be talking about. Now, the elastin is going to be the core, but surrounding that elastin core, we're going to have fibrillin molecules that are going to be deposited basically to form a capsule around those elastic fibers to help them organize and help protect them from degradation as well. So here we see our elastic fiber is at rest, and when we apply a force in these lateral directions, you can see that we can extend those quite easily. And there we see those cross links, the desmosine cross links in illustration. Now, looking at it a little more from a molecular level, and excuse the watermark, I wasn't able to actually get permission to use this, so shh, don't tell. Here we have 2 lysine molecules, amino acids, and 2 lysine amino acids, and they join together to form this ring-type desmosine cross-link. And it's because of this cross-link that's going to allow quite a bit more flexibility, but yet we are going to have a lot of rebound based into the kinetic energy, the potential energy that's going to be restored there to help snap that fiber back to its original resting position. So far we've talked about the acellular molecular components of the extracellular matrix. Now, as we move into our connective tissues, now we're going to look at the cellular components. So first, we're going to talk about the fixed cells. Now, fixed basically is a relative term. So when we look at their location, we're going to say that they really don't migrate that far away. So for most of us, Nacogdoches is our fixed position, even though we move around in Nacogdoches. Some of you may go home to Houston and Dallas. You would be commuters, transient type of cells in the human body, but for now we're talking about the fixed type. And our first cell type are the fibroblasts. These are the ones that we really do think of and associate with the connective tissue proper. They're going to be the most abundant, and they are the ones that derive the majority of what we've been talking about in regards to our connective tissue. Now, fibroblasts being derived from these pluripotential or multipotential mesodermal mesenchymal cells, they can give rise to other mesodermal cell types, adipose tissue producing fat, chondrocytes, our cartilage cells, and our osteoblast, our bone producing cells. Now, fibroblasts can have varying states of appearance, but most of the time when you see a fibroblast in a histological preparation, they're going to be active, which means they're going to be somewhat elongated, and I'm not sure that we really use the term fusiform or spindle-shaped anymore, but very tapered on the ends and rounded in the middle where you find the nucleus. And that's really the characteristic feature of fibroblasts, this large oval-shaped, dark-staining nucleus, prominent nucleolus in the very center of these with the tapered ends of the cells. We'll get an image of this in the next illustration. The other thing that's going to clue you in that you're looking at a fibroblast, especially if you're looking at a transmission electron micrograph, is you're going to see a lot of collagen fibers surrounding the cell. The cell is moving through collagen connective tissue. It's depositing this collagenous connective tissue. So that's going to be another landmark feature to look for. These cells are going to have abundant rough endoplasmic reticulum, which is a characteristic feature of cells that are undergoing high levels of protein synthesis, especially secretory proteins that are glycosylated. So it's no surprise as fibroblasts are depositing adhesive glycoproteins, collagen, elastic, they're going to have an abundance of rough endoplasmic reticulum in their cytoplasm. So here we see a transmission electron micrograph, and hopefully you're getting more comfortable looking at these. We can see the nucleus, we can see the euchromatin, this dark material just on the border of the nuclear envelope. The lighter material is our heterochromatin. We really don't see a nucleolus in this image. These parallel structures that we find in the cytoplasm, these are our endoplasmic reticulum, and you can somewhat see the alternating dark light features, which makes this a rough endoplasmic reticulum. So another characteristic we're looking for. Here we see somewhat a broad area where we have the nucleus, and you can see the tapering of the cell toward the top and the tapering of the cell toward the bottom. That is our spindle or fusiform shape. And then in the extracellular matrix, we have these elongated fibers. And although we really can't see much of a periodicity, it's there. You have to kind of use your imagination. You can see a little bit better in this field. You can see some here as well. But then we see all these little dots. And what I want you to think of is when you cook spaghetti. You take the spaghetti out, you have a bundle of spaghetti. If you look at the spaghetti from the side, it's going to look very long, like you might see here. But if you turn your hand and you look at the spaghetti on end, it's going to look like this. You'll see a bunch of dots. So these are collagen fibers that are in cross section. So you're looking at the cut round ends, and these are collagen fibers in longitudinal section. So that plane of section you always have to keep in mind as you're looking at these histological preps. Now, often histology can be almost artistic. And in this case, we have some fibroblasts that are being grown in culture. This almost looks like an epithelial squamous type of flat cell. But in culture and on plastic and various substrates, cells will attach and maybe do some different things. Here we see more of a spindle shape that we're used to seeing with a fibroblast, very tapered on the ends, more rounded in the middle, a little bit more of that spindle shape here. We have the nucleus staining with our blue color. We have focal cell adhesive contacts out here in the red staining area. And then we have our cytoskeletal components that we see. I believe this is actin, present in green. So all of these are fibroblasts, but these are in culture, and they are actively moving around in this tissue culture area. Now, fibroblasts that are inactive, they're going to look fairly similar. They may be smaller, more rounded, not quite as spindle-shaped, which are active migrating cells, and maybe less rough endoplasmic reticulum, but principally what you're going to be seeing are the active fibroblasts in these different sections. Now, this one is a little bit of a in-between, and histologists really weren't sure where to put this particular cell type. But it was different enough from fibroblast and different enough from smooth muscle that you really couldn't combine these in any one of those categories, because they have characteristics that are similar of both of these cell types. So when you look at their cytoplasmic components and cytoskeletal components, these myofibroblasts have actin filaments and dense bodies, those adhesive connections for our microfilaments, just like we saw in smooth muscle when we were in physiology. However, unlike smooth muscle, we didn't have this extracellular coating, the basal lamina, around it, and fibroblasts certainly don't have that. So once again, that is a characteristic of fibroblasts. But we find these very high densities in areas of wound healing, and even in this area where we have the periodontal ligament. So wound healing is a characteristic of fibroblast, and it's thought that maybe these cells can become either fibroblast or smooth muscle, or in areas of wound healing, they can actually come together and they can retract more to bring the new tissue that's being formed closer together, so it's not quite as daunting a task, and you won't have as large of an area of repair by pulling this together. It's, in some ways, you can think of it as contributing, much like the retraction of a clot, bringing those areas together. This is another mechanism to do the same thing. Our third type of fixed cell are referred to as pericytes. Now, pericytes are going to be on the periphery, and I believe that's where this name came from, and they're going to be around blood vessels. So we refer to them as perivascular, vascular referring to the blood vessel. And you're going to find these around capillaries that only consist of one layer of simple squamous epithelium. That would be our epithelial classification, but our identification is endothelial cells. Capillaries are only one endothelial layer thick. Now, venules are going to be larger in diameter, but pretty much they're still just going to have that one endothelial cell layer. And so around them, wrapping them, giving them a little bit of support, are going to be these pericytes. And in this scanning electron micrograph, we can see either a capillary or a small venule, and you can see these pericytes perpendicularly arranged around that endothelial layer. Now, these pericytes, like the myofibroblast, are going to be similar to two types, similar to smooth muscle, similar to the endothelial lining, and they may in fact also differentiate into either smooth muscle, full-blown smooth muscle cells, or endothelial cells to replace endothelial cells that may have been damaged. So those are the pericytes. Now, the fourth type of fixed cell, and this is the one that we probably are more familiar with than the myofibroblast and the pericytes. These are the adipocytes, commonly referred to as fat cells. And they are going to function in the synthesis and the storage and then the release of triglycerides. This is our storage form of our phospholipids and fatty acids that are going to be used in the body to produce energy. And so in our diagram, we can see that we have the breakdown product of materials we have in our bloodstream from our digestive tract. Those are going to get imported and stored in the fat cell as triglyceride, but these can also be recruited, and you can remove this material and put it back into the bloodstream as glycerol, fatty acids, and that can be plugged into our citric acid cycle to produce energy and ATP. This is just a look at a typical histological preparation, a light microscopy of fat cells. And we're going to have two different types of fat cells that we can identify. This is going to be the one that we're more familiar with in the human body, in the human adult. You're going to see mostly white space. And the reason that you see mostly white space is the solvents that are used to prepare tissue for histology are going to be organic solvents, and they will remove much of the triglyceride, leaving the plasma membrane intact, but removing all the triglyceride that was in place and leaving these large white spaces. So we're going to be looking at thin cytoplasm of cell, the nucleus pushed all the way to one side. It's going to look like a ring, and it's referred to as a signet ring, and that's going to give you the clue that you're looking at adipose tissue. Now the first type, the one that I was just describing, is referred to as unilocular adipose tissue, or white fat. Now these are extremely large spherical cells, and you really don't produce that many more fat cells as you might put on weight. You simply add in more and more triglyceride. The only way to really remove fat cells is liposuction. So in these unilocular adipocytes, fat stored in one large single drop, and that's going to give this large signet ring appearance, especially as you remove a lot of the triglyceride during your histological processing. And you're going to be left with the nucleus that is pushed all the way to the periphery, and you can simply see all the empty space. And when you see cells that have this appearance, you know you are looking at unilocular or fat or white adipose tissue. The other type of adipocyte, this is going to be much more common in newborns, in fetal tissue, is referred to as multilocular or brown fat, brown adipose tissue. The fat's going to be stored in several small droplets. You're not going to have the nucleus pushed all the way to the side. The membranes that wrap these droplets into vesicles are going to be present more after the tissue processing, which gives it the dark, more dense color. You're going to have more mitochondria in these, and the reason for the difference is the brown adipose tissue is going to be much better at heat production, 20 times faster than we see with the white tissue. But quickly, as we move from newborn or as hibernating animals, as they move into their wake periods, that brown adipose tissue is going to be lost and replaced and used as white adipose tissue. So here we can look at a brown adipose tissue. This is one cell, and within that cell we can make out the membranes of all these smaller vesicles. There's still a lot of white space, and sometimes this looks like bubbly or spongy, and that's one characteristic that I often will look for when I'm trying to identify brown adipose tissue. The nuclei somewhat are peripheral, but they're not as compressed as we saw with the white adipose tissue.